What is the difference between benign and significant genetic polymorphisms? Imagine your DNA as a long instruction manual for your body. Now, picture tiny spelling differences in that manual. These are called genetic polymorphisms. They are common variations in DNA sequences found in many people. Not all these changes are the same, though. Some are harmless, while others can influence health. Understanding these differences helps us see how genetics shape our well-being. Benign genetic polymorphisms are like small typos in the instruction manual that do not cause any problems. They are variations that do not affect how genes work or how proteins are made. These harmless changes are found in healthy individuals and are considered normal parts of genetic diversity. For example, a small change in a gene that does not alter the protein's function is classified as benign. These variants are often used as markers to study family history or population genetics, but do not requiring medical concern. On the other hand, significant or pathogenic genetic polymorphisms are changes that do impact health. These variants can alter how genes function, sometimes leading to disease. They might change the structure of a protein or disrupt biological pathways that keep us healthy. For instance, a mutation that affects a gene involved in blood clotting could increase the risk of bleeding disorders. These variants are labeled as pathogenic or likely pathogenic in genetic reports because they can explain inherited health risks. Between these two types, there are also variants of uncertain significance. These are changes where scientists do not yet know if they are harmful or harmless. Ongoing research and family studies help clarify their role over time. This helps doctors decide whether these variants are important for health or not. In family genetic testing, knowing whether a polymorphism is benign or significant is vital. Significant variants can point to inherited risks for conditions like cancer or heart disease. This information can guide medical decisions and preventive care. Meanwhile, benign variants help build a picture of a person's genetic background, but do not requiring any action. Geneticists classify these variants by looking at how common they are in the population, whether they affect gene function, and if they are linked to disease in families. Variants seen frequently in healthy people are usually benign. Those that consistently appear in people with certain health conditions are considered significant. To sum up, benign polymorphisms are harmless DNA differences common in everyone, while significant polymorphisms can influence health and increase disease risk. Recognizing this difference helps doctors and families understand what genetic changes matter and what do not.